For this final project, I will be demonstrating a simulation of E. coli path following using GRO software. I wanted to show that it is possible to simulate the directed migration of E. coli in GRO using a set of sequence signals representing food or other growth-promoting factors. The model sets the cell's growth rate proportional to the amount of food signal received, and any cells receiving a signal below a defined threshold will die. The food signals are finite and will be consumed. The first video shows the path of signal propagation. The second video shows the E. coli colony migrating along the signal path. Given that the growth rate is proportional to signal, we see the occasional overgrown cell in the simulation. As you can see, simulating the model in GRO is straightforward. The main consideration is the need to slightly overlap the signals so the E. coli can migrate cleanly. From an application standpoint, being able to direct bacterial colonies has a wide range of potential uses. You can direct bacteria to specific locations to outcompete other bacteria, or to deliver custom proteins to specific sites, or in defined patterns such as with etching. I wanted to extend the simulation with an interactive model, which turned into a game, FeedMe.gro. It follows the same simulation model with E. coli growing proportionally with the food signal received, and the E. coli die if the food signal is lower than the defined threshold. For the game, I've included a grid of emitter E. coli, which when clicked produce a decaying food signal. Once the signal decays, the emitter cell dies. The game starts with a decaying food source in the center of the grid and stable food signals in each corner. You win by successfully creating a colony in each corner. The key variables that can be adjusted are the amount of food signal and decay rate of the emitter cells, base growth rate, death rates, as well as the startup period for the initial signal are all variables in GRO.